Hello and welcome to Nikolay's genetics lessons, and today we are going to talk about genetic engineering, and here's a problem. Recombinant human insulin produced by bacteria carrying a cloned insulin gene, and now the major form of insulin used to treat diabetes. The human insulin gene encodes a messenger RNA which is only 333 nucleotides long, but the entire gene spans more than 4,000 nucleotides. There are 3 exons and 2 introns. And here's a question A. If we were to clone this gene directly from the nuclear DNA, bacteria would not be able to express this insulin gene. Explain why this is true. In your textbooks, usually you can find a picture like this. This is exon, this is intron, this is exon again, intron, and exon. So according to our problem, this gene consists of 3 exons and 2 introns. But actually, in reality, most of the genes look like this. So this is exon, this is intron, this is exon, this is intron, and this is again exon. Within most human genes, introns much bigger than exons. So non-coding sequence of the gene is much bigger, according to our problem, more than 13 times bigger than exons coding sequence of the gene. We don't need this strand, but imagine that this strand is gene, so this is DNA, this is coding strand, and from this strand we make messenger RNA. Of course, the sequence of the messenger RNA is going to be complementary to this DNA sequence. So this is going to be messenger RNA. But this is going to be early form of the messenger RNA. Later, these introns would be cut away, and we are going to see messenger RNA that would only consist only of this coding sequence. Also, it's going to undergo certain modifications. For example, here we can find 5' end untranslated regions, or 5' UTR, which also going to include 5' cap, and also such post-transcription modification will include addition of the poly A tail, which is going to be 3' UTR, 3' end untranslated region. So mature messenger RNA are going to include exon 1, 2, and 3. 1, 2, and 3. And also 5' cap is going to be here, and poly A tail would be added here. With asterisks, I'm showing 5' UTR and 3' UTR, which are not going to be translated into the protein itself, but which are needed for stability of the messenger RNA. Now imagine that this is going to be circular bacteria genome, and as you see, it only consists of regions which would be translated into the messenger RNA. It doesn't have any introns here, only exons. So if we would be able to insert such large gene into the bacteria genome, first of all, it's going to be too big, and second, bacteria just wouldn't be able to cut away introns, and also would make proteins using this sequence of the introns. And we are going to get completely different protein, even if we are going to get it, because it's going to be very large. As you see, 4,000 nucleotides stand for about 1,333 amino acids. But actually, this protein consists of about 111 amino acids. As you know, one trinucleotide code for one amino acid. So 111 amino acid, the length of this protein. So we just cannot take a gene and insert it in bacterial genome because it's not going to be expressed correctly and not correct protein is going to be made. So this is answer to the first question. And the second question, what technique should be used instead in order to get a functional insulin coding sequence cloned into the bacteria? Describe briefly how this technique works. Now here is a genius invention of the scientists. They took messenger RNA, mature messenger RNA, so mRNA, without introns, so it's already processed messenger RNA. They also added reverse transcriptase enzyme, and this enzyme we can find in retroviruses. What it actually do, it attach itself to this messenger RNA and produce DNA complementary strand of this messenger RNA. So now we have a hybrid messenger RNA, one strand, and another one is DNA strand. Then this messenger RNA strand would be degraded and DNA polymerase would make another complementary strand. So now we are going to have double-stranded DNA, which we call cDNA. C stands here for the complementary or copy of the messenger RNA. But this is going to be DNA, double-stranded DNA fragment, which only have coding sequence and doesn't have this non-coding sequence, which are introns, only exons here. And then this fragment would be inserted in the bacterial genome. So let's show it here. So it would be inserted here, double-stranded DNA, and bacterial genome also double-stranded DNA. But now it can be expressed and correct protein can be formed. So when this fragment of the double-stranded DNA, this gene which we inserted in the bacterial genome, would be expressed, so messenger RNA would be made, but this is going to be a short version of the messenger RNA which doesn't have introns. And bacteria would be able to express this messenger RNA and make correct form of the protein. In our case, this is going to be insulin. And the last question, every cell in the human body has the same DNA, so every cell has the insulin gene. However, in order to use this technique, you describe it in paragraph B, you would have to start with cells from the pancreas, the only body cells that actually produce insulin protein. Why are these the only cells that you would work? Take a look. In order to use this technique I just described, we need a lot of messenger RNA. Those we have the same complete set of genes in every our cells, but all our cells are different. Or we can say all our tissues are different. In some tissues, for example, in retina of your eye, one gene would be expressed and other would be shut down. In, for example, skin cells, another set of genes would be expressed and the rest would be shut down. So the same is true with pancreas, where we can find a lot of messenger RNA which code for the insulin, and other many genes that would be active in other cell tissues would be shut down. So in pancreas we can find a lot of messenger RNA of our interest. So we are going to use these cells and not other cells where this gene would be just inactive. So messenger RNA is not going to be produced and we wouldn't be able to make double-stranded DNA copy or cDNA of this messenger RNA. And this is all for today. Subscribe and see you in the next video. Goodbye.